Good morning, everybody. My name is Quan Zhang at the Connecticut Agricultural Experiment Station. Today is May 27th, 2020. I'm at the experimental orchard of the Lockwood Farm in Camden. And in this short video, I'm gonna show you some symptoms of fire blight. This orchard was inoculated on May 6th, so it's about 21 days after the inoculation. This is an apple variety, early Macon, which is very susceptible to fire blight. And you can see the shoots are actively growing when temperatures are above 60 degrees Fahrenheit. Usually the buds break around end of March, and after 2 months of growth, we already have lots of leaves. Flowers usually bloom at the beginning of May, and we can see that after 4 weeks we have some nice fruit set. Those are some of the healthy flowers. The crop load is slightly lower this year due to the frost damage that we had on May 9th and May 10th. As I mentioned, those trees were inoculated with the fire blight pathogens. Compared to those healthy fruits formed from healthy flowers, we also have some diseased flowers. So you can see the diseased flowers not only did not produce fruit, but also they appear as brown colored, blackened, withered flowers that also serve as inoculum to spread the disease throughout the whole tree. You can see some of the dried amantha and stigma, and then later the pathogen start traveling downward through the petal into the shoots, and then from there they can spread from these whole shoots all the way to other parts of the plants. And this can result in not only crop yield loss, but also fatal of this apple tree. A young apple tree can be killed within a year of growth. Today I would like to introduce some of our recent research updates on colonization and host entry points of fire blight pathogens on apple flowers. The infection of an apple flower by the fire blight pathogen usually takes a few days, from a healthy flower on the left until the symptom development. We can see that those symptoms are developed within those days, but what we didn't see is where did the pathogens initially colonize on which part of the flowers, and from where did the pathogens start to enter the flowers into the host, to transit from living on the plant surface to living internally in the plant. So in order to understand where did the pathogens colonize on the flowers, we need to develop a method for us to track those pathogens, track the presence of those pathogens. We used a gas reporter system, which is a gene that can transform a substrate, the X glue, into a blue color. So the blue color, later I'll show you in the rest of the presentation, will indicate the presence of the pathogens on the plant. And then we inoculated the pathogens that carrying this construct that will turn blue onto the apple flowers, and to try to detect where can we find those blue colors on flowers at the beginning, mid, and later stage of the infection. So at the very initial stage, 2 days after our pathogen inoculation, we started to see spotted blue colors on the stigma portion of the infected apple flowers. We also saw that some of the anthers, which are the tips of stamen, also turned light blue at the early stage. And on day 3, we see a lot of blue color on stigma and some blue color on some of those anthers of the infected flowers. Here is another example showing the stigmas and some of the anthers are blue 3 days after inoculation. So we further quantified how much of the Erwinia amylovora, the fire blight pathogens, are harbored on stigma and on the anther. So we used the ImageJ software to detect how much blue color are present on those two structures. The yellow numbers represent the level of blue colors on stigma, and the red colors represent the blue levels on anthers. And as we can see on this picture, the total gray value on stigma and the total gray value on anther are comparable. But the average gray color per stigma and average gray color per anther are totally different. We see that on stigma, each stigma, there are probably about 4 times more bacteria than on anther on average. However, because a flower has only 5 stigma, but they usually have up to 20 anthers, the total amount of the blue color or the pathogen presence, if you add them all up, would be approximately the same on stigma and on anther. So in the amantha, there are amantha shells and also the pollen grains harbored in the amantha shells. So we further evaluated whether only the pollen grains or pollen grains. Here is the amantha shell that we dissected from infected flowers. You can see some of those blue dots on the bottom, which are the blue-colored pollen grains. And when we look at the anther under a light microscope, we can see that only the blue colors are only restricted to those pollen grains, but not necessarily the anther shells. We also use a different method to label the bacteria with green fluorescent protein. And we can see that those green-colored pollen grains are also observed when they are attached to the stigma. Later, we also found that the pollen grains can serve as vectors to introduce Erwinia amylovora to the Flora cup. As you can see in this picture, there are lots of those blue dots, which are the pollen grains on the bottom of the Flora cup. We also saw some of the pollen grains that, after released from the anther shells, are attached to the style of the stamen, dropping downward to the bottom of the Flora cup. These observations suggest that pollen grains could be an excellent vector for the dissemination of the pathogens from the original site of infection, the anther, down to the secondary infection site, which is the Flora cup on the bottom. Another observation we made during our investigation is that the petal fall junctions are excellent pathogen entry points and colonization site. We constantly saw those blue-colored concentrating near those petal fall junctions. Here is a healthy flower with some of the petals still attached and some of the petals already falling off. Apple flowers' petals often fall during later flower development when the pollination already occurred. Once the petals fell off, those petal fall junctions could represent as excellent injuries or natural openings for the pathogens to invade. We saw some evidence that even before the petals are fallen, there are already some of the bacteria attached to those areas and are ready to invade. Sometimes we saw blue pollen grains at those petal fall junctions, suggesting that the source of the bacteria could be from pollens from the anthers. Occasionally, we would also see apple flower petal turn blue, especially the edges of those petals that has those trichomes present. As the progressed infection of the flora cup, we can see the flora cup are turning blue, and also the epidermis started to peeling off, which is a sign of infection. Sometimes we also saw the petal fall junctions turning blue, in addition to the flora cup. So in summary, we saw that both anthers and stigmas are sites of colonization of the fire blight pathogen. Although each stigma harbors significantly more pathogen cells than each anther, the total amount of the pathogen cells harbored on each structure are actually comparable. In anthers, the pathogen cells are mostly on pollen grains, but not necessarily on anther shells. And we also saw that the pollen grains can serve as a vector to introduce the pathogen cells from the original site of infection from the anther down to the flora cup, where the infection occurs. It may also serve as a vector for the pathogens to be spread between flowers by pollination insects. And finally, the petal fall junction are also important pathogen entry points to blossom blight that was overlooked previously.